Okay, so hopefully your notebook is loaded by now. If not, no big deal. Just give it a second. You can follow along as soon as it loads up. So as Tim mentioned, I'm going to be talking a little bit both about SAM and BAM files and some how to begin to pull information out of them, how to parse them. So in bioinformatics, there's lots of different types of read mapping software that is always being developed or new versions, but the format for the actual mapping files has been fairly consistent. SAM format, which stands for Sequence Alignment Map, is one of the most common file formats that you get out of a lot of aligners. So all a SAM file is just a tab delineated text file. It has 11 mandatory fields. Header fields or header lines are denoted by an @ symbol. I'll show this example in a second. And so while SAM files are human readable, they can get to be quite large, especially if you're working with very large sequencing data sets. So an alternate to the SAM is the BAM file. I have here, it says binary alignment map. I think that's wrong. I think it's BGZF is what the B in BAM stands for. I meant to change that. But it is a compressed file. So unlike SAM, it's not human readable, but they're much more compact and easy to work with. So most pipelines use BAM over SAM, and that's mostly what we're going to be doing today. So one suite of tools that exists for parsing SAM and BAM files is the Samtools. So Samtools has a ton of functions. It's it seems every time I go and look it up, I find more things you can do with it. And if you want to really dig into them, there's a link to the manual page in the document. However, I'm going to walk us through some of the most commonly used functions of Samtools. So first off, just to describe a general pipeline for read mapping, if you've worked with mapping data before, this will be quite familiar to you. And it's kind of complicated. It could be its own topic of discussion, setting up a read mapping pipeline. But just in general, once you have your mapped reads in the BAM SAM format, such as output from BWA, bowtie, whatever, there's a few steps you need to do to process those BAM or SAM files before you can start downstream analysis. So firstly, you usually filter your data. Typically, you remove unmapped reads from your file. I'll talk in a little bit exactly how to do that. You then will sort your file. So both by config and scaffold or scaffold and by coordinates. So we do this using the Samtools sort command. So I've already, for the example BAM file that I have, I've already sorted it. So we don't need to run this again. So I'm just showing you the syntax of it. So it's Samtools sort. This -o is the out file you want to create, then the file that you are sorting. And then finally, after sorting it, you want to create an index of the BAM file. So this is required for a lot of programs downstream. And also it speeds up when you're searching a BAM file. So we can actually go ahead and run that. So what this will do is Samtools index creates a bam.bai for, BAM index. So you can see I created it in this data folder. So if you look over on this side of this tab right here, you can look in the data file or the data tab. If I run this, oh wait, no, sometimes it gets disconnected from the kernel. If your commands aren't doing anything, by the way, you can check this right at the screen here. It should say bash. If it doesn't, be sure to select it. So if we go ahead and run this, it should generate an index. Let's just make sure. Yeah, see, there you go. It creates this .bai file. So now that we've sorted and indexed it, to actually visualize the alignments, one of the most useful tools in the Samtools toolkit is Samtools View. So this allows you to, as the name suggests, view the contents of a BAM or SAM file. And by default, it outputs it in SAM format here. So if I go ahead and run this, just a reminder, I'm piping this to the head command, which just displays the first 10 lines of a file, so it's not printing the entire thing. So if we go ahead and run that, You can see this is what a SAM file looks like. So it's a little intimidating. I'll walk us through quickly what each of the columns are, each of the fields. So as I mentioned, there are 11 mandatory fields. I've listed what exactly they are right here, but the first is the read name. The second is the so-called Bitwise flag, which I'll talk about in a second. The third is the name of the reference. So this is the config that this read maps to. In the little toy example, we have the chromosomes are just named chromosome 1, 2, and 3. So this is chromosome 1. The fourth column is the leftmost position that that read maps to. The fifth is the map quality score. There's more details on how exactly this is calculated in the manual for Samtools, but or about SAM files, but just briefly, it's an expression of how good a map is and how unique it is. The next column is this, it's called a cigar string. These can get a little complicated, but what this contains is basically information about any indels the read has, if there's like clipping of the alignment, things like that. So this tells you where in that alignment, if you have them, any indels occur or whatever other feature. These next three columns we can skip. They're not super important. This column contains the sequence of the read, as you can probably tell. And then the final mandatory column contains Phred scores for that read. So each position here corresponds to a base in the sequence. And then you can also have some optional fields after it, which is what these guys are. This kind of depends from mapper to mapper. So by default, Samtools View will print out all the alignments. However, if you have specific regions you want to look at, you can provide that. So for instance here, if I run this first line, I want to look at just the first chromosome, let's say. So I could run that line right there. So remember the scaffolds here are called chromosomes, are just called 1, 2, and 3. So this is just saying print all the reads that map to the first chromosome. You can get more specific here if you want and specify a subregion of a scaffold, or you just have this colon and then the coordinates for the region you want to pull out. You can see that here. So that is useful if you have just, say, a specific region, like one in particular. If you have numerous regions of interest, what you can do is you can format your regions as a bed file and pass that to Samtools View. So bed files, if you're not familiar, is just a type of annotation file. It's a simple tab delineated or tab separated list. So just this little example one I have here, we can print what that looks like. You can see it's just a 3-column list. So the first column is the chromosome. The second and third are the start and the end. So this bed file is just saying, so the first 100 bases of each of the 3 chromosomes in this BAM file. So we can pass that bed file to Samtools View with this -L command right here. Run that. So I had it printing to a different folder called just subset.sam here. If you look in the data, you should see it right there. I won't bother opening it. One thing to bear in mind when passing a BAM file to Samtools is that it can be rather slow if your BAM file is quite large, because for whatever reason, this doesn't use the BAM index to search, so it can take a little while. One other thing to note with this command, in addition to the -O option for setting the output, is this -H. This option is important to remember as it adds a header to the output. So if you remember from above, the header of a SAM file is delineated by an @ sign. And these are important because a lot of downstream tools that you'll use require a header to parse these files, and it'll give you an error if you don't. So always be sure when you're printing to a file to include the header. So Another thing you can do with Samtools View is convert between SAM and BAM formats. So depending on what you're doing. So as I mentioned, by default, Samtools View will output in SAM format. So converting from BAM to SAM is super easy. You just run Samtools View, add the header, then the out file, and then your input BAM file. Oops. So if we're going from the reverse, from SAM to BAM, all you have to do is just include the -B option. You can see here, -B, add the header, the out file is the BAM file we want, and the input is the SAM file. Okay. And again, generally, you're going to want to work with BAM files because they're just more compact. So one of the fields in a SAM file that I want to talk about in particular is a SAM flag. So this is the second column. It's the Bitwise flag. So this concept can be a little weird or a little difficult to grasp. The flag value is an integer, and it's the sum of a series of other integers, decimal values, that give you information about how that read maps. So here is kind of a list of what each of those decimal values are. So for each of these that is true, that read will have that flag. So for instance, if a read has a flag of 99, what this corresponds to is a flag of 1 plus a flag of 2 plus a flag of 32 and a flag of 64. So if we look what that means that read is mapped along with its mate. So that's the 1 and the 2. And it's in the proper orientation. So its mate is on the reverse strand, and this is the first read in the pair. You don't worry too much about like memorizing this or whatever. There's whole websites out there that will explain what a SAM flag is. You can just search SAM flag, and you can input it and it will tell you. The only really flag that is probably worth memorizing is this number 4 right here. So which corresponds to a read that's unmapped. And so as I mentioned, most often you're going to want to filter out your unmapped reads. The way you can filter using Samtools View is either with the lowercase f, or which only takes reads that match a certain flag, or the uppercase f, which is its opposite, reads that don't match a flag. So if you wanted to get rid of all the reads that are unmapped, you'd use the uppercase F4. We go ahead and run that, just showing the header of it. This will filter out anything that has the 4 flag in it. So it's worth noting not just things that literally have a 4 in the 4th column, but also, for instance, 77 and 141, which I think corresponds to the reads are paired and both of the pairs are unmapped. So you can filter on any criteria using the other flags, obviously. So for instance, if you wanted only reads that map in the proper pair, you could do the lowercase f, which is get only reads that have this flag. But I think if I run this, it doesn't actually give any output because, so this BAM file we're using is a subset, so none of the reads are properly paired, so that's why it doesn't give any output. But you could use this for like a proper BAM file, obviously. Okay, so now that you have done all those initial steps, I want to mention a few things you can do in terms of downstream analysis. So one of the most common things you want to know from a BAM or SAM file is the coverage. So how over your assembly or whatever that you mapped to. This is because if you have 0 coverage or very low coverage in a particular region, that can affect your confidence. Like maybe in the assembly, maybe it was misassembled here. If you have really low coverage over a region, this will affect the accuracy of your snip calls, stuff like that. There are many ways you can calculate coverage and depth. I'm just going to show you the ones in Samtools. If you're familiar with another tool called Bedtools, you can do it that way too, but we're not going to go there. So one option with Samtools is Samtools coverage. So if we go ahead and run this, what this creates is a table that has for each contig or scaffold in your BAM or SAM file, just different useful summary statistics. So these are the different chromosomes 1, 2, and 3, the start and end position of the chromosomes, how many reads map to each of those, how many bases are covered. Coverage, so that's this one, refers to the proportion of bases that are covered. So this is not like a super well covered region, it's about half. So half the bases have a read over them. The depth, the average depth. So again, this is quite low because it's only a subset of read, and it's near the start of a chromosome, so it's like 0.1. Then the base quality scores and the map quality scores, the average. So similar to Samtools View, if you can look at just at specific coordinates, though note the syntax is a little different. You need this -R flag here. So this is looking at just chromosome 1. If we run that, you can see it just has the stats for the first chromosome. If you want to visualize coverage, just like really quickly, just to kind of get like a fast sort of impression for how your reads look, you can do the -M option with Samtools coverage, and this will create a histogram of coverage over the contig. So if we go ahead and run that. So we're looking at just the first chromosome, the first 1,000 bases, so base 1 to 1,000. You can see here it creates this little histogram. The position is along here, the percentage of bases that are covered in each window, which I guess is 25 by default. Some different summary statistics about it. This isn't, you wouldn't like publish a figure with this, but if you just want to get a quick kind of read on your data, you could do this. You might also want to look at per base coverage rather than the average coverage. So for this, you can use Samtools depth. So here, if we go ahead and run this and look at the first few lines. So you can see this creates a just a 3-column tab delineated text file here. So the first column, so again, we're looking at just chromosome 1, position 1 through 1,000. So the first column is the name of the contig, so it's just chromosome 1 in this case. The second column is that position, so this is each base, so base 1, 2, 3, 4, 5, etc. And the third column is how many reads are piled up over that base. So you can see here it's all 0, because this is at the start. So this is saying that at the start of our contig, there are no reads that pile up here. So the file, so you can pipe this to a output. This format can be quite useful for other downstream analysis. So you can import this to say R or something like that. And you can use this to plot like a histogram showing the distribution of per base coverage. You could make a histogram showing the distribution of depth over contig, kind of like what we did with the Samtools coverage, but much prettier looking. Yeah, so that's mostly what I use this command for Samtools depth, for importing it to R or whatever. Okay, one other useful kind of summary that you can do with Samtools is using the Samtools stats command, which, as this name suggests, gives you a variety of quick summary statistics about how your reads have mapped. It generates a lot of information, most of which probably isn't going to be useful to you. So we're just going to look at the summary numbers that it generates here. So the command is just Samtools stats. I'm piping it to grep to just pull out certain lines. You don't have, and then cut out the second column of it. You don't have to worry about this too much. It's just making it not super verbose. So if we run this, you can see you get all the information you could want about this. How many sequences are in it, average quality scores, how many are mapped, how many are paired, average fragment length, lots of different stuff. Okay. So I just have two more little things that Samtools can do. Again, it has a ton of functions. You can check out the manual page if you want. I encourage you to do so. I always find new things. So one neat trick you can do is, so if you notice the BAM file, it has all of the sequence information about your reads. So if you say want to then pull all of the read data out of your BAM file as a FASTQ file or FASTA file, you can do that with Samtools FASTQ or FASTA. We go ahead and run that. You can see it is outputting this as in proper FASTQ format. This can be handy, say, if, I don't know, you're short on space or something like that. You don't want to have like your raw reads files just taking up room or something. You can get rid of your raw reads files, and if you ever need them, you can just get them out of the BAM file like this. I also should note, if you have paired end data, there is an option to output the reads to different output files, so pair 1 and pair 2. Then finally, one last trick you can do is you can use Samtools to merge BAM files with Samtools merge, which can be obviously useful if you have multiple rounds of mapping and you want to combine them all together. They have to be sorted first. And you can have as many BAM files here as you want. If you don't specify, the headers will be merged, and also they'll be interleaved properly. So as long as they're sorted, like the sort order will be maintained in the merged file. Okay, yeah, that's about it. That was rather quick, but hopefully it gives you some idea of how to begin tackling these files. If anyone has any questions, I'll be sticking around for a little bit. You can either message me or just you can talk in the channel. And yeah, if no one has any more questions or whatever, you're free to go. Thanks for stopping by.